Hello students, today we are going to start with the biomolecules, different type of biomolecules. We have already completed the introduction part. In the introduction we have studied about the definition of biomolecule, what is biomolecule. Second, we have studied about the different type of tests to identify the organic and inorganic elements. We have done the acid test and the ash test. Then after that we have seen the tables. Okay, three tables we have seen, and that we have also studied the primary and secondary metabolites. So we have completed that introduction part in 2 videos. Now after that we'll start the biomolecules. So the first biomolecule we are going to study, okay, that will be protein. So in the protein, what we have the monomer, and that monomer is amino acid. Okay, so first of all, before understanding the proteins, we have to study what is amino acid. Amino acid is actually is produced in our body. We can synthesize, or some of the amino acid we have to take it from outside, from different food sources. So in the NCERT it is given that amino acid is called as the substituted methane. Okay, so why it is called as the substituted methane? The reason, whenever you see the structure of methane, you know CH4, right? So here this is the structure of methane. So here what we have to do, we have to replace this 3 hydrogens. One of a hydrogen will be present in one side. This will be acid, carboxylic acid we will add. Here we will add the amino group, and here we will add the variable group. Okay, then this structure will be the structure of the amino acid, general structure of the amino acid. Now what is this R, or variable group? Okay, so this is the general structure of every amino acid, right? So depending upon this R, we can have different amino acid. If suppose this structure of amino acid, in this R we have hydrogen, that will be the simplest amino acid, that will be glycine. In this variable group, then if is in the variable group we have one acid, that amino acid will be acidic amino acid. Then if it is base, then it will be a basic amino acid. If it is a sulfur containing amino acid, aromatic amino acid, different type of amino acids we can able to get in this. Okay, so just see, whenever you see this structure, general structure of amino acid, then you can see here in this same alpha carbon. Okay, this is the first carbon, here this is the second carbon with which all the structures will be attached. So this will be called as the alpha carbon. So in this alpha carbon, in one side we have the acid. Okay, this is the carboxylic acid here, and this will be the base. Okay, so here if in this R we will add one acid, then there will be two acids, so this will be an acidic amino acid. If in this variable group R region we have another base, okay, then there will be two bases and we will have a basic amino acid. So this is the general structure of one amino acid, right? So remember this thing, what is an amino acid? This is the structure. Okay, then after that we will see what actually the amino acid form the bonds. Okay, so let us see, whenever it form different bonds, see how it actually form bonds. Okay, so see, first of all, suppose this is one amino acid, we will draw another amino acid here. Okay, this is the first amino acid, and here we will see the another amino acid. Okay, this is the another amino acid. Now in this, okay, we will have a bond. So how they will have the bond? You can write this structure as this, right? Carboxylic acid, and here you can write like this. Okay, now what happens here, we will have the bond that will be called as the peptide bond. So here one water molecule will be repelled outside. Okay, and what bond we will have? This bond will be called as the peptide bond. Okay, then what will be the structure now in this dipeptide? Because two amino acids will be there, so the two amino acid attached to each other by a single peptide bond, so this structure will be a chain, right? So the chain will be like this. Here we will have the bond. Okay, so this is going to be the bond. Okay, so this will be what bond? This will be the peptide bond here. So this is going to be the peptide. So in the amino acid, if you see this way, we can have more amino acid can be attached here, will repel this water molecule, and this way the bond will be formed. So whenever you see a chain, okay, suppose one chain we draw, okay. So let's just see what will happen in this chain. Okay, so in this chain, suppose this is one amino acid, this is the second amino acid, then this is the third amino acid, this is the fourth amino acid, this is the fifth amino acid. So they will be attached to each other by the peptide bond. In one side we will have a base, okay, this will be a amino group, and in this side, on the other hand, on the right hand side, we will have the carboxylic acid. Okay, so in this structure, what we can see here, this structure will be a sparingly soluble solution. If you add it in the water, then one part of the chain will be soluble in the water. Suppose this alcohol part will be soluble in the water, but this acidic part, this is a long hydrocarbon chain, so that's why it will be not dissolved in the water. So it will be half soluble, half insoluble. So it will be sparingly soluble in the water. So you can see a long chain of a peptide, okay, or long chain, or you can see a protein. In this protein structure, one side will be a base and another side will be a carboxylic acid, right? So this will be the structure, how the biosynthesis of protein occurs. One amino acid attached to another amino acid by a bond. That bond will be called as the peptide bond here, with a replacement of water molecule. So if one water molecule is removed, then we can have the peptide bond. And if you add water, then the amino acid can be detached from each other, break the peptide bond, and we can have the individual amino acids. So here up to this, what we have seen, the structure of amino acid and how amino acid makes bond, right? So after that, let's just see the next part. What will be the next part? That if you see this amino acid, it can form Zwitter ion. Okay, so what is the concept of Zwitter ion? See, now Zwitter ion concept you have already seen in the chemistry subject. So in this amino acid, what happens, you see, first of all, you just see a amino acid. Okay, so this is the carboxylic acid group, okay, and here it is the amino group, here it is the hydrogen, and here it is the variable group, this is R. Okay, so if you take a solution and that solution have a pH, suppose acidic pH, okay, so in this acidic pH, what will happen, one hydrogen will be added, okay, so it will be converted into this, right? So what happened in acidic pH, this amino acid will show a positive charge. Now the another pH, you can see, suppose this solution of pH, this is suppose a basic solution, okay, this is a base solution, then what will happen, it will take hydrogen, it will take a proton, right? So here what will happen, we will have this amino group, here we have the carboxylic acid group, here this is hydrogen, this is the R. So what will happen in this basic solution, in the basic pH, what will happen, it will take hydrogen, it will take a proton from it. So that's why what will happen, hydrogen will be taken from here and we will develop a negative charge. Okay, so here in this carboxylic acid group, we'll have a negative charge here. Okay, so you have seen that in the acidic pH, amino acid develop the positive charge, and in the basic pH it will develop a negative charge, right? Now there is a very interesting thing we will see in a solution. If it happens that in a solution, suppose this is a solution of an unknown pH, so we will take this pH later. Suppose what will happen here, we will have this carboxylic acid group, okay, and here we will have this amino group, here variable group, here hydrogen. So at a specific pH, what happened, a hydrogen, actually a proton from this carboxylic acid group is coming and it will be added into the amino group, okay. Then what can happen, you see, here in the same ion we can get both the two charges, that is positive as well as the negative charge, okay. So this structure, this ionic state in which a compound develop both the positive charge and the negative charge, this is called Zwitter ion. And that specific pH, due to what compound develop the positive as well as negative charge, that will be called as the isoelectric point. At a specific point of pH, when a compound develop both the positive as well as negative charge in the same compound, then this will be called as the Zwitter ion. Okay, remember this will be the Zwitter ion, and that specific point of pH when this Zwitter ion forms, okay, then this will be called as isoelectric point. Okay, so isoelectric point can be can be written as pi, and we have seen in chemistry we have to find out the pi of a solution. We have done a number of numericals in this, and if you are not getting it, you will get it later to find out the pi of it. Okay, so yes, this is about the Zwitter ions. So after that, let us move to the next part, the different types of amino acids. Okay, so what are the amino acids, the different types of amino acid? If you see, then what type of amino acid we will see depending upon the availability in our body. We can have three types of amino acids. So we will see the types. First of all, we will have the essential amino acids. Okay, we have semi-essential amino acids and we have non-essential amino acids. So what are the differences between essential and non-essential? Essential means you require the amino acids in your diet. That means your food should contain this type of amino acid. Why we require in the food? Because our body cannot synthesize that amino acid. That will be called as essential amino acid. And non-essential amino acid is not like that, it is not required. We don't require it in our diet. But why this happens? Because our body can synthesize the amino acids by the different essential amino acids. So that will be called as non-essential amino acids. Essential for our body, but we don't require it in the food. Why? Because our body can synthesize by ourselves from the essential amino acids. Now, in the question it comes, find out from the from four amino acids, find out the find out the essential, or among this amino acid, which one is the non-essential amino acids, right? So what we have to do, you have to remember any one of it. Okay, either you can remember essential, all of the essential amino acids you know, then you don't have to remember the non-essential amino acids. So I have a trick for it. How many essential amino acids we have? Yes, we have 7 or 8 essential amino acids. Depending upon the person or lactating mother, we can vary the essential numbers of essential amino acids. Either it can be 7 or 8. So I will write here essential as well as the as well as the essential and semi-essential amino acids together. So you have a trick. First of all, you can remember like this: PVT, TIMHALL. Okay, so this will be your essential amino acids. First of all, this will be P for phenylalanine. Next one, V for valine, T for threonine, then T for tryptophan, I for isoleucine, M for methionine, next one H for histidine, A for arginine, L for leucine, L for lysine. Okay, so among these 10 amino acids, 8 of them they are essential because we cannot synthesize by our own. We have to get it from the outside food sources. From this two, this arginine and histidine. Okay, sometimes it can be threonine also. So HA, remember histidine and arginine, these are the two semi-essential amino acids. What do you mean by semi-essential amino acids? They are produced in our body, but it require more time, maybe 10 to 12 years. It can be slowly produced in our body. So this will be called as semi-essential amino acids. These two of them, generally histidine and arginine, they are called as the semi-essential amino acid. And rest of all, whatever you get apart from this 8 amino acids, this 10 amino acids, all of them that will be non-essential, like aspartic acid, glutamic acid, serine, cysteine, okay, cysteine, glycine, that will be all non-essential amino acids. Suppose I can give you a example. Suppose it is tyrosine. Okay, so what is tyrosine? Tyrosine is one non-essential amino acid because from we can produce phenyl, and from phenylalanine we can produce tyrosine by a process of deamination. Okay, if you remove one, phenylalanine have one amino group, so if you remove this amino group by deamination process, deamination occurs inside the liver, and we can produce tyrosine. And tyrosine is the precursor of number of hormones in our body, like the thyroxine, T3, T4, melanin, adrenaline. This can be produced from the tyrosine. So tyrosine we can synthesize by ourselves. So that's why it is non-essential. But from where we have produced this non-essential amino acid, it will be from the essential amino acid. So I hope you remember, understand what is essential and what is non-essential amino acids. So once again, what are the essential amino acids? The amino acids which we can produce by ourselves from the different essential amino acids, that will be called as non-essential amino acids. The amino acids which are synthesized in our body very slowly, that will be called as semi-essential amino acids. Two of the semi-essential amino acids we have seen, that are histidine and arginine, and the essential amino acids are those which we have to require in the food, and we cannot synthesize by our own. Then this will be called as the essential amino acids. What are the essential amino acids? That will be PVT-TIMHALL, that is phenylalanine, TIM, what are they? Phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, leucine and lysine. The rest, that is histidine and arginine, we can produce in our body very slowly. That will be called as the semi-essential amino acid. Okay. After that, let us see that the other different types of amino acids depending upon what is present in the variable group. I have already told you in the earlier introduction of this starting of this video that if this variable group is an acid, then it will be acidic amino acid. If the variable group is a base, then it will be basic amino acid. So depending upon that, we have number of amino acids. What are they? It can be acidic, it can be basic, alcoholic, neutral, sulfur containing, heterocyclic and aromatic type of amino acids. Yes, you have to remember it is important. So first of all, acidic amino acids, you can remember it very easily. You will have acid there. What are they? One is glutamic acid, another is aspartic acid. So Z and A, they are acidic amino acid. You can remember it easily, glutamate and aspartate. This glutamic acid and aspartic acid, so we can it is produced in our cycle, urea cycle. Here we can have this. So these are the acidic amino acids. Next on, basic amino acids are also very important. H This will be La, lysine and arginine. They are the basic amino acids where we will have an NH2 or another base added to the variable group. Next one, alcoholic amino acids, where we have one OH hydroxyl group there. We have serine. Yeah, structure of serine you have to remember, it is given in your NCERT. So structure you have to remember. Next one, threonine, it is also alcoholic amino acid. Next one, neutral. On the neutral, this will be very much important. Glycine is very much important because the structure is given in your NCERT and it is the simplest amino acid. Why? Because in the variable group we have the hydrogen. So this will be called as a neutral amino acid. Next, in the neutral amino acids we have valine, alanine, glycine, leucine and isoleucine. That will be the neutral amino acids. Then after that, sulfur containing amino acids. They are going to be very much important because here the sulfur containing structures you get in the NCERT book. In the second year you will see the structure of antibody. Here it's a peptide. It's having more than 50 amino acids, so it's a long chain. So that it is amino acid chains will be attached to each other by the disulfide bond. Another structure we have, that will be the structure of insulin. In this insulin structure of also we will see two chains, and those two chains, one is A chain, another is beta chain. A chain attached to beta chain, or alpha chain is attached to beta chain by disulfide bond. So that's why these two structures are going to be very much important. So you remember this thing. This will be CCM. Okay, cysteine, cysteine and methionine. These are going to be very much important sulfur containing amino acids. You just, wherever I make the star, you have to remember it. And next one, heterocyclic amino acids. They are proline, histidine and hydroxychlorine. Next one, we have aromatic amino acids which are essential. We have seen it, not tyrosine, but phenylalanine and tryptophan. They are the aromatic amino acid. So I have told you that phenylalanine is a structure essential amino acid from where we can produce tyrosine. Right? So here these are the charts of different types of amino acids. You have to remember the structure of amino acids. But is it all of the amino acid you have to remember? No, you have to only see whatever the structure is given in the NCERT. You have to remember, you have to practice it. You just draw a general structure. You already have seen it, that one alpha carbon, we have one side carboxylic acid, one side we have amino group, one side we have hydrogen, and only the variable group is going to be changed. Right? So from the NCERT book you have to see whatever the structure of amino acid is given, you have to see. Like serine you have, glycine you have, these structures you have to see. And from here you may be asked one type of question, from the following amino acid you have to find out the odd one. So suppose 3 of the amino acids will be given from neutral side, one will be the acidic amino acids, or one may be the basic amino acid. You have to find out the odd one, that means the basic amino acid, right? So that's why this will be important. So that is all about the amino acid you have to know. Then after that, later on in the next video, we will see after this amino acid what will be the next polymer will be formed, that will be protein. So after that we will see the protein. So that's all about the amino acids. I hope you understand. Thank you.